Well, wonderful. Hi, everyone. Welcome to the special edition of the Hox Zodiac, Invoking Your Animal. The Hox Zodiac is a long-standing collaboration it's an art science collaboration between the pig, Victoria Vesna, an artist, and the sheep, myself, Siddharth Ramakrishnan, a scientist. And it melds the Chinese zodiac with genetic manipulation, foods, and myths and mythology. And we ask you to contribute your recipes and stories and myths to our YouTube channel and also to the Instagram at Hox Zodiac and also on our Facebook page at Hox Zodiac again. Um, in this special edition of the Hox Zodiac, each month we are invoking an animal of the Chinese zodiac, and we start with the rat. And we as human animals are constantly collaborating, creating, and controlling other species around us, either as pets, as food, as science experiments, or even weave them into our myths and stories. But we tend to forget them and we tend to forget about them constantly. And hence the constant problems with things like climate change, plastic pollution, deforestation, and of course, industrialization. So with this monthly series, we hope to better empathize with our relation to our animals and creatures around us, and better define our place and responsibilities in this planet. So let's begin by introducing ourselves. Victoria.
Yes, so I am a pig, and if you go into our Hox site, you will see that my celestial animal is a yellow dragon. When I first heard I was a pig, I was a little disappointed, but the more I discovered about it, the more I felt really grateful to be this animal that is so giving. And the sign is water. My color, the element is black. And the every animal also has a ruling organ, and that's sanjiao, which is a kind of a general nervous system organ in Chinese medicine. And then if you look at herbs and food that we've compiled together, this is what's recommended for the pig. So if I come to the table, I either offer pork, which would mean that I offer myself to the table, or I offer what's recommended to me as a pig by Chinese medicine, Ayurveda, and then looking at what Western medicine would offer. So we also have this idea of science detox connecting to the particular organ. And then if you look at scientifically, we could just go on and on. So every time we do this, we will focus on an animal. And there's so much to be said. We could spend all day talking about the rat and how much it contributes to our well-being, in addition to our fears and myths, etc. Siddharth? Oh,
Thank you,
maybe.
Victoria. And I am a sheep and I'm also a scientist. So I don't know if in the Chinese parlance, sheep and pigs are scientists or artists, but it's going to be interesting to find out. My heavenly creature is also a yellow dragon. Maybe that's our cosmic connection, Victoria. Both of us are yellow dragons.
Yes.
And the sheep rule the small intestine. And it looks like there's another sheep or a goat who is joining us along with Danielle. And yes, so this as a scientist, this kind of collaborative project has really opened my eyes out to different ways of approaching how we talk about animals, especially within science experiments, and how uh, we kind of lost the touch of being a naturalist as a scientist is something that I've been discovering more and more as we're going through this project. And today we have a special guest, Kathy Hai, who is an honorary rat because of her work. So I'll let Victoria introduce her.
So if you could stop sharing, sort of go to the horse. Uh, Kathy Hai, who I've known for many years now, we really go way back, is a horse, and an earth horse no less. So that's really wonderful because I'm an earth pig. But I'll just mention, I'll show her sign to everybody because it's really beautiful that she's joining us because her ruling organ actually is the heart, and that makes sense. So here we are. Thank you, Siddharth. So we have the vermilion bird is her heavenly creature. And people always ask, well, what does heavenly creature mean? It means that in our lifetime, in our physical body, whether it's a horse or human, we're subject to a lot of karmic happenings and changes. But the celestial creature is really celestial. And a lot of the Chinese zodiac got lost in this idea of the earthly definition of the animal and superstitious ideas. In fact, the very way that it came together was by looking at the celestial planets and then seeing a horse, let's say, connecting it to different aspects. So the ruling, even though Kathy is earth, the ruling element of the horse is fire. And it's a very yang element, and it looks to the south. The taste is bitter. So when you look at Ayurveda or Chinese medicine, that's what we're looking at. And the planet is Mars. So welcome, Kathy.
Thank you, Victoria. This is great. I hadn't known some of these things about like the vermilion bird, which is just amazing. So this is so wonderful. Thank you.
You're welcome. So welcome. You know, when I saw your piece with the rat, I just, I don't think I ever looked at rats the same again. And I have to say, it did very much influence thinking about this project as well, because you shifted the perception of that animal, and that meant so much to me to really take something that we just think is dirty and brings plague and is nasty and in labs. And Siddhartha also talks about the rat a lot because as a scientist, he has to be in a situation where he's dissecting and, you know, literally sacrificing the rats for research. So your work bringing the rat in, and I actually pulled it up, so I'm sure you're going to talk about it, so I won't do it twice. Siddharth, did you want to say something about the rat?
Yeah, so we constantly think about rats as Victoria mentioned, like dirty creatures, but we don't realize how smart they are, and social animals they are. And now more and more research is going into how much empathy they can show each other. So if a rat can find another rat who is in trouble, they actually go out of their way to help each other and kind of pull them out from, say, when they're in the water or something like that. So they actually are super social creatures. And we don't, even though scientists know this, they tend to house them separately sometimes because it's easier to maintain. And so it's kind of, we are manipulating them in ways in which they're not supposed to be manipulated. Uh, my first experience with rats came from when I was a young biology high school student, and we used to do dissections. And because we didn't have great lights in the lab, we did them out on the corridor, and we used to expose the circulatory system of the rats. And there used to be a crow shoer who used to make sure that the crows don't come and pluck the rat circulatory system and go away. And I remember one of my friends forgot that he was a crow shoer, and the crows actually came and like plucked the open rats and like just like had a feast. And so he got yelled at by the teacher saying, your whole job is to shoo the crows away. and, and he forgot about it.
And
And
of
that
course,
was
the
my first.
crows are so smart too, right? I mean, they're just amazing.
Yeah, the crows are really smart. If you actually notice, the way they dissect roadkill is they actually find the orifices, and they're actually able to poke their beak and then like kind of do a section through the animal, just like you would take forceps and scissors and cut through them. The crows are able to find that like weak point and then cut through it.
True
It's really
scientists.
fascinating.
So, so Kathy, I just want to do a short introduction to the audience. Maybe you're new to some people. To me, you've always been around and you're so amazing because you've moved from so many different worlds with technology, art, biology. You collaborate, you really work with medicine, and you've received so many awards, Guggenheim, Rockefeller, NEA, et cetera, many museums. You also were at Symbiotica, which for those that are not aware of is in Perth, Australia. That's an amazing biotech and art lab that started way in 2000, I think. So working with bioart. And then recently, I actually had the pleasure to not collaborate, to introduce with you this wonderful new initiative, the Nature Lab, which is in the Sanctuary Environmental Education Center out in North Troy. And we'll have a link to that. It's actually an amazing, wonderful community science lab, and it's open to the public. So as someone who's a professor, you also are very much interested in taking education out to those who don't have access to this kind of education, and to really raise awareness around biology, technology, art, equity, anything that's really bringing humanity to a lot of this work that tends to be in closed lab doors and not accessible to a lot of people. So when you did the embracing animal, I was just so amazed to have that live camera on these rats, and it's withstood the test of time, I must say. Even though the site looks like it did, I feel like conceptually it is so strong that it's still there for all of us. And we also linked to your wonderful, wonderful essay, Playing with Rats, that's a tactical biopolitics book, Art, Activism, and Technoscience, that is edited by the late Beatrice da Costa and Kavita Phillips. Highly recommended to everybody. I think it's an important way to back up a little bit of what you're going to share with us today. So with that, welcome, welcome, my dear friend, and with admiration, I introduce you.
Oh, thank you so much, Victoria. That was such a beautiful introduction, and it's such a pleasure to be here with you and with Siddharth and everybody else who's here, which everybody's looking very beautiful, so welcome. So yeah, I mean, I know that this project has been talked about a lot, and so many of you on the call may be very familiar with it, but as we're honoring rats, that I was trying to think of what to say and what to talk about today. And I thought that I would go back to my dear friends, the rats, who were my colleagues for a few years. And I thought that maybe I would read one of the pieces of writing that I wrote 15 years ago that's on the website. So you can find it there, called The Politics of Empathy. And it is about my relationship with the rats. It's very personal. And while I'm doing that, I would love to share some sort of home movies we made together, just of them hanging out. I mean, I'm not really, I'm the spy eye, but that's so that you can see me through the camera. You can imagine me on the other side. But we became quite close. So let me see, is it possible for me to share my screen?
Yes. Yeah.
Okay, cool. And I think I'm just gonna do it this way. Oops, wait, do this.
We see you.
So let me get this up.
There we go.
Okay, cool. So there's a little bit of, there's sound in this video, and I'll talk about the sound afterwards, but you're hearing a couple different soundtracks. So that's just going to be the background to me reading this, which won't take very long, and then we can launch into conversation and such. So this is the politics of empathy. These rats are named Matilda, Tara, and Star, Echo, and Flowers. They are all transgenic gene transfer rats, HLA B27, transgenic rats to be precise, exhibiting a phenotype similar to humans suffering B27 rheumatoid arthritis-related disorders. They've been microinjected with human DNA that sets them up for a precondition to be autoimmune challenged. The injection of human genetic material occurs in the pronucleus of the mouse embryo, and it's passed generation to generation ever after. They're prone to develop diseases like reactive arthritis, psoriasis, inflammatory bowel disease, and other things. They're developed for pharmaceutical research studies in systemic inflammation. Matilda, Tara, Star, Echo, and Flowers are also retired breeders, meaning they are used to give birth to baby rats that carry their added gene. They came from a laboratory that breeds such rats and mice too, and sells them to researchers. Their ears have holes and yellow markings on their fur as a kind of numbering system. These marks denoted their identification, their names, until they came to live with me. I bought them to try and make them live as long as possible and to see if they could become healthy given their prior genetic conditioning. I will treat them holistically with alternative medicines such as homeopathy, environmental enrichment, and also good food and play. Stress is one of the triggers for their conditions. I know because I too have autoimmune problems in the form of Crohn's disease, corticosis, and others. Thus, I identify rats and feel as though we're mirroring each other. I feel a great kinship with them. And when I see them feeling tired, I recognize that kind of exhaustion. I know they need rest in a way that is total. That's my dog Lily, by the way. If they ache when they're touched, I understand this is from fevers. I also know they don't know how to behave as pets. They're not pets. They're extensions, transformers, transitional combined beings that resonate with us in ways that other animals cannot. The combination of animal and human is traversed here with a kind of commitment and lack of choice. To empower just a few, to give them a retirement they earned, why not take back these parts of ourselves and repay the care? They're on exhibit here to introduce them and this research to you, the public. This special laboratory has been developed to extend their observations of humans and to extend their lives, and also to invite you to assist in this work. I'm getting information about them from an interspecies animal communicator who has been checking in with them. She speaks with them telepathically and asks questions for me. She said, "The rats don't have a lot to say, but what they do say is very strong. They speak to me as a group. They say that they are very happy receiving all the love and care you give them. They like humans and are studying them as well." Matilda, Tara, Star, Echo, and Flowers, I need your help and suggestions. How can we make your lives more enriched and your health better? If you have an autoimmune problem, what do you treat yourself with? What foods do you, the public, enjoy, and what do you avoid? I'm not against the kinds of scientific research that works with animals, but I do
No, you're just eating so well. It's
but I
really
do
good.
think that there are other kinds of
Yeah.
research to be conducted. We have injected human materials into them. We have a real kinship with these rats, and still they are forgotten workers. The transformation that is taking place here is astounding and delicate, and we need to exercise a kind of empathy that results in understanding how we use them. The politics of caring of such a forgotten creature, a pest, a disposable one, has to have a transformative effect. And this is a kind of play that you'll see that Matilda and I used to do when she came home from Mass Mocha after she had finished her job in the gallery. So while this is playing, the other thing I'll just tell you is that the kind of high frequency sound that you hear in this soundtrack is a downsampled ultrasonic track from a project that I've been working on for over 10 years called Rat Laughter. And rat laughter is a combination of many ultrasonic giggles of rats. It's at a certain frequency above our hearing range. I was lucky enough to be gifted these recordings from a scientist, and we were able to, as a team, Michelle Temple, Matt Wellens, and Eric Rothenberg were able to work with the ultrasonic soundtracks, clean them up, and layer them so that hopefully we'll be able to play them to lab rats, and maybe they'll find some enjoyment listening to a laugh track from other lab rats. Just gonna skip ahead a bit. You can see that we could play this for hours. She really loved it. So I think it's good to spend time with the rats. So that's what I was trying to do with you today. And so I hope you got a little sense of how wonderful they were, how much they taught me, and how much they enjoyed their retirement. Thank you.
So, what happened to them? Are they still alive, or when did they pass away? How did they pass? Tell us
No.
about that.
Yeah, so rats typically don't live that long. Rats
Mm-hmm.
generally live about 2 to 3 years. These rats, because they were, autoimmune compromised, and bred to be sick really only were supposed to live only about a year and a half. They actually lived pretty like to about closer to 2 years, which was longer than the scientists who had designed them originally had predicted. So they kind of did have a long life for them. Echo and Flowers were part of an original uh, resident show in Boston at a commercial gallery. They were on exhibit for a month, and then they came home with me and lived with me for the rest of their lives, which was, as I said, closer to 2 years. I didn't get them until they were about 10 or 11 months each. So we didn't have that much time together. This is Hazel, by the way. Hazel would like to say hello to everybody.
Hi,
Hi,
Hazel.
I'll take off her bird collar. And so then when I was asked to be in the Mass Mocha show, I was able to get Matilda, Tara, and Star. They were on exhibit for 10 months in the gallery. There were apparently, tens of thousands of people who saw them. The gallery claimed that they had 10,000 people per month. So that means like they saw 100,000 people met them. And at the thing that was great was we didn't change their circadian rhythm, so that the person who really fell in love with them and worked with them was the night watchman, Mike Weber, who really just was taken with them. And when I brought them all home after the exhibit, Mike and his wife came and asked me if they could take them home with them. So actually, Matilda, who you saw playing at the end, died with me before she went. The other two went with Mike and Peg Wilbur and Weber, sorry. And they kept Star and Tara until they died with them months and months later. So it was a really, really beautiful story. They have kind of love story.
Yeah. Tell us a little more about the rat laughter frequency. That sounds amazing.
Thank you. So that project has been, like kind of my backbone project for years. It was started at Symbiotica when I was there, and that was really wonderful to be able to do the beginnings of it. But that was back in 2009, 2010. Since then, a lot of research and work has been done on the technology of ultrasonics. And so we've come very far with that. And so it's been a lot easier to do to find the kind of recordings and actually work with them in the time that's passed since then. Originally, it was very hard to isolate the recordings. Rats laugh at this frequency of about 50 to 55 kilohertz, which is above we kind of max out at about 20 for our hearing. And they laugh when they're kind of tickled or they're playing with each other in a giggle. And I tried to collect giggles on my own with my wonderful artist friend and PhD students, Michelle Temple, who became a collaborator with me on this project about 3 or 4 years ago. And we went to people's houses and tried to tickle rats and record their sounds. Eben Kirksey, an anthropologist who's now teaching in Australia in Melbourne, also tried to tickle rats and record their laughter, their giggles. So we've had many attempts. We got about 4 seconds of ultrasonic giggle out of all that. So luckily, this scientist who had been working with an amazing person whose name was Jake Pansep, who was one of the people who first sort of started publishing about rat giggles. Sadly, he's passed away. He passed away a couple of years ago. But one of his collaborators was able to, agreed to bequeath me these recordings, which are really dirty and, noisy. And then the technologist that I was working with, Matt Wellen, was able to clean them up and make it possible for us to hear them by kind of doing this kind of downsampling. And that's what you were listening to. And we have an ultrasonic speaker ready to go with the recording. And I'm ready to get back in the lab as soon as I can get back into things after
Did
since
you
the
hear about
pandemic. It's been a little slow.
Did you hear about Deep Squeak?
Yes, Deep Squeak is amazing. Yeah, Deep Squeak is you want to talk about who they are? They're
No,
really cool.
go for it. Yeah.
They're a group in University of Washington who have been doing this incredible project of kind of mapping algorithmically different registers of ultrasonic conversation with rats, between rats. And so that eliminates all this kind of noise that we were talking about. It eliminates so much of that so that you can hone in on just a very particular frequency. Their recordings are fantastic. They're really truncated because they really just look for that one little frequency echo. And if you listen to how we talk or think about how you hear sounds, they kind of have a way of rising and falling. And so it's not quite so precise, in my opinion. And so this kind of having the whole thing made it possible for us to really add a little bit of the ambience between the laughters. But Deep Squeak is amazing. They're doing incredible work. I love those guys.
Tell us how you got the idea to work with the rats, and what was it that prompted the whole thing?
Um, well, two things prompted it. One was the work of Eduardo Cac and the Alba Bunny, which I'm sure everybody's probably aware of that project. And if you're not, check it out. It's really awesome. It was a way that Eduardo was trying to also work with transgenic animals. And the bunny, Alba, was in a lab in France. And it had been Eduardo's goal to take Alba out of the lab and bring her home. But that was really difficult. Again, at the time, this was in 2001, 2001.
Mhm.
And so that was really hard to do at that point, also because Alba was in France and Eduardo at that point was in the US. So, I felt that was something that was kind of too bad about that project because I know that had been his intention. He also raised some great questions about how we should consider these transgenic animals and really consider the care of them. So that was really important to me. And then on top of it, I was reading a lot of Donna Haraway, and particularly her Modest Witness text, where she talks about mice, the onco mouse particularly,
Mhm.
that produces cancer tumors being our sister because of this transgenic nature of that
Mhm.
model animal from science too. So those two things kind of spurred me. And then I guess there was one third element. Adam Zaretsky was working at RPI with us at about the same time that, I was thinking about all this. So he was a great, how do I call it, a cheerleader.
Mhm.
Whenever I came up with an idea and said, I can't do this, he goes, you certainly can. So without Adam, I don't think this project would have actually come to fruition.
So
Really
you
amazing.
mentioned in your video as you were reading, you were talking about how these some of these rats were used to breed, and then they would then sell the babies to researchers,
Yeah.
and it kind of reflects on how most research is being done in, especially in rats, is you put an order on an online form, and then the rat is delivered to you, and you don't know what happened before, you don't know how they're raised, you don't get to play with them as you did. So there's a certain sense of remoteness, and the animal becomes a tool in the science lab as opposed to an animal.
Right.
And I can you comment on that a little bit?
Yeah, no, those are great observations. Thank you. That you're absolutely right. So these rats that I had, as I said, were retired breeders. So they were not young. You could get pups, as you were mentioning, shipped to you. I chose the retired breeders because I was a retired breeder at the time. I thought that was appropriate to have my kin that reflect that. But not that I ever really bred successfully, so that's not quite the same. But anyway, but they had tried to get into that facility, and it wasn't that. It's really hard to get in. Plus, as you said, these animals are then used for different experiments, and they're just considered kind of like you would consider ordering, I don't know, a chemical or some kind of cells that you might want to work with for an experiment. You hope that and expect that the chemical and or the model animal, like a rat, would be kind of the same each time you get it, so that you can successfully continue to work on your experiment itself and not have that much aberration between the results. And so that's a very controlled situation, and that's why these animals are bred the way they are and are sold the way they are. And and the work that I did with them was completely kind of the antithesis. It's not a controlled experiment. It was not anything that you could predict the outcome, nor could it become any kind of statistical results, because it was not done except for those 2 iterations. And so the other thing is that, as you said, since these animals are used in research, I find that they're also not tracked. Like, we don't really know how many rodents are used, and this includes mice now too. Mice and rats are used in scientific research like around the world. It would be great to do a massive mapping project to really count the numbers, because I think that their numbers are in the billions,
I think that's such an important point. And if you dial back 1 year, the whole pandemic started in the year of the rat in China, and right
Yeah.
next to the animal market that was right next to the biotech lab. And once the pandemic became from epidemic to the pandemic, then the billions of rats maybe have turned into trillions. I mean, there was
Yeah.
suddenly all the regulations, everything just opened big time, even for monkeys and other animals as well, to get the vaccine, through kind of fast track. So it would actually be very, very interesting to see that and how much they contribute. Which brings me to Siddharth a little bit, because we have over the years had so many conversations about him as a scientist in the lab working with rats. And I always had 2 questions that maybe Siddharth you could share. One is, do the rats really reflect us? In other words, if something like for arthritis, for instance, works on a rat, would that work on a human? Are we that similar? How about the neurons, etc.? And what does that really mean? And the second one is, how does a scientist have some kind of empathy when you have to daily do this kind of work? And you were even mentioning a story with a student that was quite interesting.
Yeah, the first question is interesting because there are some aspects of animal biology which is shared, like the fundamentals of neuroscience. But there was this amazing article in the New York Times about how this one scientist, a lot of work on sepsis, on how organ failure happens because of sepsis was studied in rats. And this one scientist kind of felt like the bacteria that the rats have and the bacteria that we have are very different. And so the sepsis conditions cannot be the same. And he kept trying to say that the way we treat sepsis in humans is wrong, and we need to have a different way of treating it. And every time he tried to publish, the reviewers said, well, you have not shown this to be true in rats. So they kept kind of asking him to go back and show that his research worked in mice and rats. And they didn't, because they eat something completely different. Their habitat is different. And he just could not get his work published. And so finally, I think he just published it in some like not a super high impact journal. And finally, they came to show that the way humans react to infections is very different than the way rats react to infections. And so there is a time and a place to use certain animal models, and for other things you shouldn't. So almost like following this rote, this golden thing of the rat is the most important thing for a human research is not the way to go. So that kind of shows you, like, as you are asking, is the rat the best model for human research? The empathy, I think the bit when Kathy was talking, I was actually thinking, um, this might be something that we have to introduce in undergraduate labs, where students interact with the animals. And so right from a very young age, when they're developing their scientific minds, they learn about more about going back to the roots of naturalism that I was talking about, and are trying to understand the animal for the sake of the animal instead of thinking about the animal as a tool. And the way to develop that empathy is, I think, doing this kind of play interactions and trying to work with the animal. When I was a PhD student, I worked on snails, and I was fortunate enough to work in a lab where um, my my mentor used to, when he made tomato something chili at home, he would kind of bring all the tomato peels and feed them to the snails. And the mango, and then, of course, the tanks would get super dirty and we had to clean them, but still this, you could kind of see which ones the snails enjoyed, which they didn't. And a lot of our times we just spent like looking at these snails like swimming around, and they're super fast if you actually think about it. And so I think that level of observation, and which kind of was emphasized in Kathy's video, of how most of it is just observing. And that observation of animals is kind of lost now, I think, because you just pick them up on the day of the experiment, do whatever you want on them, and then you put them back in their cages. And maybe an animal handler or a animal care technician is taking care of them, but the actual experimenter or the science research person is not. And I think that divide needs to be broken down. And the other important thing that Kathy was mentioning was this transgenic bit. You can now get a transgenic line of rat for anything. The plastic industry has a whole transgenic line of rats which are resistant to estrogen. So they can kind of prove that plastics don't have any effect. Whereas if you go to different labs where you don't have those specific lines, then they'll show you that plastics have effects on animals. So it's kind of really easy to manipulate this on that fine genetic stage, basically. So.
And it's very similar to food industry. If you think about the animals and the industrialization of animals. So when we do this project, people sometimes misunderstand that we're against people eating animals. No, it's really about the treatment. So even if you eat the animal, how do you understand, or how are you grateful for that? So Siddharth, you also talk about how in the lab you were at once dissecting and the animal actually was alive, and it was like traumatic to you.
Yeah, that was a traumatic experience. I was a part of my job was to remove 18-day fetuses from a pregnant mouse, for a pregnant rat. And so you actually place an order saying, I want a rat delivered at when she's 18 days pregnant. And then you get them, and then you're anesthetize them with carbon dioxide. And then you are working on them. And then really sadly, in the middle of my dissection, I guess I did not anesthetize her properly. She woke up, and I panicked and I didn't know what to do, in the sense, the anesthetic, the way you anesthetize the animal is you put them in a chamber and then you give them carbon dioxide, and you couldn't do that because she was already on the dissection table. And it takes a lot of effort to put them down. And that's something that you realize quickly as you're dissecting animals, is it's very easy to open them up. It's very difficult to close them up, or make them recover. It's much more difficult to put everything back in place again. And I had an undergraduate with me who was super excited by all these things that were happening. It was like this amazing experience. And I kind of had to like tell him, this was not an amazing experience. This was this traumatic event, and he needs to feel sad that this had happened to this animal on this dissection table, and it should never happen again. And almost teach them that process of trying to understand the pain of that animal, or whatever experience it was going through, and empathize with them with that a little bit. So that is something that, again, that I think we need to talk about, some of these failures as scientists, or even as humans, and failures when we're working with animals, that we tell the next generation and the students so that they understand that, are able to empathize with these animals better.
Well, I also discovered that you cannot use the word sacrifice in scientific papers, right?
Dolores has really interesting comments on in
Can
the
you,
chat.
why don't you read it out? Can we bring
So
in Dolores into
Dolores
this?
was working on nude mice, immune deficient, and all shipments came with a sign, life handled with care. Our protocols indicated we could not use the mice for experiment before a cooling period, so their stress levels were lower and their environments were enriched and they were treated as well as possible. There are more and more papers that are out there now which talk about enrichment of the lab animal and what all they need to be provided in order to have a good, even fish, um, in order to have like an enriching experience in the lab. So that's a good turn of events that's going on, I think, on some level.
Yeah, I would like to say also, I appreciate your saying that. Yeah, I sat on the animal care committee for our university for a while, and you know, that scientists are genuinely interested in this kind of enrichment and trying to make sure that there's not those kinds of harm happening. But it's also difficult because there's also expectations for research to be produced, and there's a timeline, and then people are going up for tenure, and you know, publishing is important. So there's all kinds of pressures on scientists, I think, too, to negate this kind of you know, care and empathy that we're talking about. So I think it's really a complicated situation, for the animals for sure, but also for the scientists, you know. And I don't think all the scientists I've ever worked with have been incredible with their animals. You know, they've been really cognizant of what's happening with them. But it's just there are lots of limitations, I think, within the systems that we built. So I think it's a big systemic issue that we need to look at.
I think part of the problem is coming from the way this is being projected to the public. And there's been more and more pushback saying you're getting public money in order to do your work. What is the benefit for humans? And instead of talking to the public about learning about the animal for the animal's sake and the value of that, and that's kind of been lost in the messaging. And so when I started about 20 years ago in the sciences, even within a span of 10 years, I could see in the neuroscience, where about 30,000 people meet for neuroscience meetings. Initially, there was 70% animal research, just talking about animals and what they do and all that stuff. And then at some point, it just shifted towards how does this relate to humans and how is this human centric? And a lot of that is because the public money that's coming in was saying, kind of shifting is to, you need to have this focus. And that's a problem, I think.
So, Dolores, are you able to join us and speak? Because I think what you're writing here is really interesting, your research with the mice. I'm not sure if it's possible the way we have it set up. All right. Well, we'll just mention that in your case, you needed to keep the mice alive for a longer period as you were watching their tumor burden, the number and size of the metastases. So they had to be healthy, quote unquote, after injection, but also after each and every IVVM session when they were alive, anesthetized, and with their livers under the microscope and blood flow was recorded, fluorescent cancer cells watched. Wow. I mean, it's just incredible how much these animals are contributing and how it's invisible in a way. And yet at the same time, they're so present in our daily lives too, especially in cities, but also in the country. There's different types of rats. I mean, they're just part of our ecology. And in London, for every person there's 5 rats, they say, but we don't see them. And during the pandemic, I heard that because of the restaurant closures, the whole ecology about the rats going into the trash got disturbed, and suddenly the rats were hungry and getting out on the street. So the invisible became visible very quickly. Um, Clarissa joined us from Fortaleza, and she wanted to make some comments, but there's a protest outside currently that,
I can share
oh, good,
briefly. Yeah,
great.
I'm sorry if it's too noisy because we're having
It's
a
fine.
protest outside.
We hear you
Uh,
fine.
and I'm happy to join, and thank you, Victoria.
Welcome,
Thank you,
Clarissa. It's always
Cedar.
great to
Thank you,
have you.
Kathy, Danielle, and all who
Tell
joined
us
in.
a little bit about the protest first.
And so the protest is against the president. One more, yeah.
Good.
Let's see, it's not a violent protest. Mostly people just expressing they're not happy and see what we can, if we can have an impact.
Mm-hmm.
That's it.
Mm-hmm.
And so after the Zoom, I'll probably be joining.
Good. As
It's
long as it's
mainly
not
about,
violent.
yeah, it's more like a pride. Just people walking with posters and some people talking and expressing their opinions. That's it. And so, since you were talking about rats in street and cities and how they are around, but we can see them, especially during the day. Yeah, I was considering bringing to the rats, just as I said in the form, a fresh tomato or an organic fresh tomato, since people still nowadays used to try or consider killing or the rats using poisoned tomatoes. So they really love tomatoes, and it's funny that Siddharth was saying that as a young scientist, he was feeding the snails with tomatoes as well. Yeah, and I was searching online for maybe people were just not doing this nowadays, but yes, and sometimes they commit mistakes and they eat the poison themselves.
Oh
And
my
I
God.
found a couple of cases in India, in Brazil.
Wow.
They cut the half and they give the half to the mice to kill them, and parts of the poison stays in the hands and they eat and they die. Yeah,
Wow.
it's tragic. But I found an interesting paper. It's not recent, it's from 2013, but a scientist was discussing this issue from an ecological point of view. And the problem is not only poisoning the rats or the mice, especially if you're considering growing something in your garden, not only
Mm-hmm.
tomatoes, they will come and they will feed and they destroy your fruits and your, vegetables. And people just insanely keep delivering these pesticides. Yeah.
Mm-hmm. Mm-hmm.
And birds, when they eat the mice, so they die.
Mm-hmm.
And now we have second generation of pesticides, and they kill in a very different way. And so if you have time, I want to read just a small. Uh,
Yes, go ahead.
well, every year, a huge number of birds are victims of second generation anticoagulant rodenticides used by exterminators, farmers, and homeowners. They're found in such brand names as Deacon, Hotshot, Generation, Talon, and Havoc, and they sell briskly because of our consuming hatred of rats and mice. The most pestiferous species are alien to the New World and therefore displace native wildlife. Both first and second generation rodenticides prevent blood from clotting from inhibiting vitamin K through the second generation products built to higher concentrations in rodents and are therefore more lethal due to anything that eats them. The difference between the first and second generation of pesticides. The second generation was developed by Imperial Chemical Industries of London at the request of the World Health Organization, because rats appeared to be developing tolerance to warfarin, a first generation rodenticide. It's a very good idea. For a rodent to get a lethal dose from a first generation rodenticide, it has to eat more than once. But that's not a problem. Leave first generation baits out for a week and they're just as efficient as the second generation. What makes second generation rodenticides so non-selective is that they kill slowly. So rodents keep eating them long after they are ingested. They have ingested a lethal dose. By the time they expire or they die or are about to, they contain many times the lethal dose and are therefore deadly to predators, scavengers, and pets. That's
It's
it. World Health Organization helping the world.
so, you know, what's interesting about that, when I lived up in Topanga, we would have huge problems with people poisoning rats because, you know, first of all, they're field rats. And of course, then the snakes and the whole ecology just went out of control. But what's interesting about your story is the warfarin part, because after a heart surgery or any kind of heart patient, they actually give warfarin to people now. So like tiny bits of warfarin, which literally is rat poison. So that circle of poison as medicine is just, to me, mind blowing, actually. Yes. Thank you for that contribution, Clarissa. That's so cool. Yeah, so warfarin is part of it. And if we look at what's recommended for rats, people in the sign of the rat, their ruling organ is the gallbladder. And so they're really recommended to eat high protein foods, but to stay away from deep fried foods and anything heavy. So lots of fish, of course, cheese. It's almost like a stereotype with the mouse and the rat. Lots of vegetables, fruits, and dates, low level grains. So this is the kind of food that here we of course have the tomato, which is something that the rats love. Um, so the idea is also to offer some suggestions to people with their sign. And what we found, which we don't even understand so much how it happened, but over time, as we incorporated some parts of Chinese medicine, and a lot of it we dropped because it's so foreign to us, we don't even understand some of the different herbs or vegetables. We looked then through Ayurveda, through Siddharth's background, we pulled it in, and that was through taste. So the taste for the rat is salty. And so we looked at vata, which is an element in Ayurveda, and salty, and looked at that food. Then we went to Western medicine and looked what's recommended for people who have problems with the gallbladder, usually stones. And we compiled a kind of a list of ingredients, and we find that a lot of times people would say, this is actually a food that I really like. And somehow I think we hit on a pretty good menu for the rat. When you come to the table as a rat, you don't offer yourself usually, but I will say that people do eat rats. And there's nothing wrong with that. Even that was survival. And still, we're talking about usually rats from the rice fields. And they're food just like any other animal. And a lot of, in fact, in China, in certain parts of Asia, during the times of terrible famine, this is how people survived. So again, there's some gratitude to rats also for our survival in just a very basic way.
I think Sam wanted to share something.
Yeah, please.
This is my first time joining you, so thank you for having me, and thanks to Danielle for having me over.
Welcome, welcome.
Thank you. So I thought I would share a story about rats in Hong Kong. So I lived in Hong Kong for 5 years and just recently moved back about a year ago. And while I was there, I learned about what they call the Third Plague, which happened in the late 1800s in Hong Kong. And what happened was, like, early in the 1800s, Hong Kong began to be colonialized by the British. And as that happened, areas where local Hong Kong people were living were starting to be gentrified, and Hong Kongers were pushed up into the higher elevations and more dense populations. And their living environments were not very great. And so a plague began, and it wasn't officially recognized until a few years after it began. I think it was recognized in 1894. And at first, inspectors would come through neighborhoods and just gut these homes where there were like multi-families living in like very small rooms. They would take everything out, put it in the street, and burn everything. And then eventually, as they started to learn more about the pandemic, oh, I have a visual aid actually. Should I share my screen or just put the link?
Yes, you can share your screen.
Okay. So the bottom photo here is a photo of some of the burnings of belongings of local people. So they would bring it out into the street and just burn it there in the street. And eventually they started to attribute the spread of the plague to rats. And so they put these rat bins, here's a photo on the bottom left, on the post and on the street. So when anyone found a rat, they were to put the dead rat into the bins, and twice a day an inspector would come around and collect all of the dead rats and remove them. And this is how they tried to manage the plague. The upper left photo is another photo where you can see just how integrated the rat bins were into everyday life. This is just a local market seller, but eventually the story of the plague is really fascinating, and it's not, totally, centered around rats specifically, but there was a really, actually, I don't know, because you guys are scientists, so you might know something about this, but there was a big controversy about there were two leading scientists who were trying to find a vaccine or create a vaccine for this plague. One was from Japan, and I don't remember where the other one was, but there was sort of a competition, and one used the other's research and ended up getting it published and got a lot of notoriety and became like the savior of Hong Kong from a scientific standpoint. But he actually used the Japanese guy's research to do it. So anyway, it's not a terribly well-developed story, but I thought it was a really interesting addition to conversations about rats. And Victoria, what you were saying before about the ecology of rats and how, how we coexist with them and how that changes in certain circumstances, I think is really interesting, and this might be a relevant addition to that conversation.
That's so interesting.
It's so fascinating. And I have to say, this is our 13th year of working on this project. We actually started in the year of the rat, and that was not, a conscious decision. It just happened. But it's never stopped. The stories never stop. The amount of stories for each animal is just magnificent. When you come to mythology, personal stories, dreams, food, all kinds of stereotypes, fears. I mean, there's so much part of our existence, and to just incorporate that into our thinking immediately shifts our way of coexisting and understanding that we're animals, we're part of this larger ecology, and that's just so important to do right now. But I'm just amazed every single time. It's never a repetition. There's always something new we learn, something new we add. And we do it over the table, usually with food. And the food is very much about us eating, offering either ourselves or whatever is good for us. And that brings again the whole issue about food and how do we treat food itself, not only just the animals, what do the animals eat and so on. So thank you for adding that because it's completely new to me. I don't think Siddharth heard of this
Yeah, I haven't
either.
heard of it, and it's kind of a nice kind of circle back to Hong Kong because that's kind of where the whole Hox Zodiac table started. We started in Hong Kong in
That's right.
2011, so it's kind of interesting how it circled back to that. I want to ask one thing Kathy mentioned in her piece was how uh, injecting human DNA or genes into these to create these transgenic rats. You were talking about that, and now there are these human brain organoids that are now injecting into rat brains. So almost human mini human brains are being injected into rat brains. So do you have any thoughts about that? Is that more of a transhumanist thing that's going on?
So there you have a great picture of that. Can you pull it up easily?
Kind of,
Kathy, it's on you to answer that one.
Oh, cool. No, that is so cool. I've only worked with colloids, and so I haven't worked with brainoids before, but I think that it's an amazing idea. I mean, it is really kind of freaky, like freaky, but it's no more freaky than the work they're doing with other animals growing organs
mhm.
in them for human use, such as pigs and things like that. So I mean, it never amazes me what research is being done because it's something I could never even imagine. But I don't know, how does it work? I mean, how does the organoid still keep the profile of wherever the original brain cells were coming from? Like if it was yours or mine, would it be our little brains going into that rat?
Yeah, that's a big question, isn't it? Like they are saying that the organoids can form connections by they're testing the organoids on in a tissue culture thing. They actually form functional connections, and it looks like when you put them into rat brains, they're actually forming connections in the rats. But
Wow.
whether the rats are thinking human thoughts, maybe we'll never ever know, right? Because you can't really
Yeah.
communicate on
No,
some level.
it's a whole nother level of communication, I think, happening there. That's quite amazing. I mean, as sort of horrifying as it is on the one hand, it could lead to some very interesting outcomes. So we should also have mini rat brains then implanted in us. So just to be fair.
That's kind of funny because the one way to test rat learning is this thing called a Morris water maze, and the way that works is you put a rat in a pool of water and rats don't like to be in that pool of water, and there's a little hidden platform, and the rat has to swim around to find the platform, and once it learns where the platform is, it'll kind of go back every time, and unless the rat has a memory impairment. So this scientist has created a virtual Morris water maze for people. So so you wear this thing and then you are kind of dunked in a pool of water and you have to kind of go around and find the platform kind of.
That's fantastic. That sounds great. You must share that with all of us. That's great. Thank you.
So on another note, Dolores is sharing that there are two words in Romanian, that's her background for rat, sobolan and guzgan. And when Anna was a baby, she used the diminutive of guzgan, guzgane, as an endearing term. And as babies look like tiny new rats, so fresh, so curious, so active. And that actually, thank you for that, Dolores, because it reminds me of an animal model to think about, which is the naked mole rat. This rat lives in a hot hypoxic environment in Africa. And its brain, speaking of brains, can survive without oxygen for a very long time. And it does not feel pain at all. It lacks pain receptors. So, of course, you kind of need oxygen to feel pain. I just find it really fascinating that this creature is out there that can survive without oxygen. So, yeah, no, there's so much to learn still. And unfortunately, part of what drives us in this project to continue is not this endless genetic engineering that we're witnessing and messing around with animals and with ourselves as well, is the species, the disappearance of species at such a speed. I mean, we just can't even follow it, and it's frightening. In fact, it really is to see how species are disappearing at such a speed. Yeah, it's not so good. Siddharth, what were you sharing?
So we're
Oh, go
we're back
ahead,
to
Clarissa.
yeah, no, I'm saying that now. So we're back to politics.
Yes, yeah, exactly. I mean,
If
it
you
really needs to change. Our behavior needs to change. And it, comes from the top in this time. So bottom up is what people want, but top down is what we're getting. So
yeah.
it's kind of a contradiction in
Yeah, but
rules.
at the same time, the top down is we the bottom up is feeding the top down. Yeah,
Yeah.
yeah,
No, but the tide is turning. I actually feel that the tide is turning, and you can see it by the floods and the fires, and just nature just is really just getting red flags, red flags. Nobody's paying attention. Okay, it's just gonna get stronger until we really do have to wake up. At least those on the top, they have to breathe the same air we do, eat the same food we do. So there's some basics. We can survive without a lot of things, but not without air. Um, Now, what's interesting also is I don't know how many people are familiar that there's rat temples in India, which is pretty cool. If you just Google, and I did it for the audience here, you can see all of this is around rat temples, and you literally are seeing rats being fed milk, and people coming in honoring and worshiping the rats. Of course, this would be in India where every single creature ends up having a sacred element. It's kind of hard to believe, but there it is, and it's just pretty amazing actually if you think about it. So it's called Karni Mata Temple. I don't know if you know
I've been
anything more
there.
about it.
I've been there.
Okay, do tell, do tell.
So it's in the northwest of India. I studied in the northwest of India, and I've been there, and it's pretty amazing. And there are also people also put out grains. So there's all these like sweets that they do with laddus and like and milk and things. So that's also out there. And then they also put out grains, but the rats don't touch the grains. They go for the heavy, fatty food, which is full of sugar. And the pigeons are the ones who are eating the grains, and the rats are the ones who are eating all the. And then they say if there is an albino white rat, the other rats just get rid of it.
Oh,
So
really?
it's very rare to see an albino white rat in which is kind of predominantly what's used in the science labs, right? And so these rats don't like them, so they get rid of them. So
So what is the general framework for the temple to the rat? What does it mean? Do you know?
I
We
don't know
should
the
do a little
mythology
research.
behind it, but
Okay, I found it. Give it
okay.
to Google. So I admit it's on the spot. The Karnimata temple in Deshnoke, and I'm probably mispronouncing it, apologies, about 20 minutes from Bikaner, Rajasthan, holds two main legends spun around the mysterious worship of rats. According to one, a 20,000 strong army of fled battle and went running to the Karnimata temple. The temple spared their lives but banished the cowards to live in the temple as rats. Thankful to not have been sentenced to death, they vowed to serve Karnimata for eternity. The other legend has it that Karnimata's stepson drowned in a lake while trying to drink water. Karnimata then sent the god of death to revive him. The god of death granted a special gift to Karnimata, allowing all her male children to be reincarnated as rats. There you go. So there's in a temple there could be as many as 20,000 rats, it says, and the visitors are prohibited from harming them.
Unrelated to it, we did the way why we went to the temple is we did go to Bikaner and from my college, and Bikaner has the best deep fried parathas you can eat.
So next time we do a dinner honoring the rat, we'll have the parathas.
Yeah, deep fried paratha. They're not just paratha; they're deep fried
Even though
parathas.
it's recommended against for the rat to not eat deep fried food. You're supposed to eat the fish and light proteins. That's really hilarious. The other thing I just wanted to mention briefly is for the cookbook that we're working on, Monica Lucasio in Vienna is actually our collaborator, and she is a rat. And the only reason she's not here is because she is doing this at the Mediterranean, having a nice dinner or lunch with her boyfriend Max. And she's eating the right food. So she wanted to share. I can't be there, but I am eating what's prescribed. Say hello to everybody. So hello, Monica. Monica's a fabulous rat, and she is doing an amazing job for the cookbook that we're putting together. She's also a great artist that works with a lot of trauma and different mycelium, kombucha, body, all kinds of beautiful stuff. So rats are really significant, and it's not by accident that they're the first on the wheel of the zodiac. They're actually considered the smartest animal of all the 12 animals of the zodiac, and certainly can survive everything. So we honor them today. Any last thoughts from our guests here? Somebody's asking, Dolores is asking if the albino rats maybe smell different, which is why they would get attacked. Who
Yeah, that's a good
knows?
question. Yeah, I don't know. I don't know if they can see that well. Like, I don't know what aspects of their physiology would be different.
Mm,
They don't see as well as other
Yeah.
rats. Their sight is very foggy, very
mhm.
blurred in general. So
Do they
they're
have pink eyes?
they do.
Mhm, mhm.
So they're sensitive to sun. So they I mean, I'm sure, I don't know if they would smell differently. That's a good question, but they certainly probably behave differently,
Mhm.
and they're probably more
Mhm,
cautious and things like that. But I'm just guessing.
mhm. Wonderful. Well, Kathy, thank you so much for your work and your contribution today. We really, really enjoy it. And Dolores, you're a rat. That's amazing. I didn't know that was your sign. I wish I knew. You would have so been highlighted. I guess we did it in some way. So uh, we will add you to this and certainly have the best we could. Um, so Clarissa, enjoy yourself and put your voice out because it's important. And thank you for joining us. How wonderful to have, always wonderful to have you. And Colette, Sam, Bobby, nice to see you guys. Danielle, So
good
good
to
to see
see
you
everybody.
always. Yeah, so great to see you. So we bid everybody goodbye and give us your hands. Ciao.
Bye.
Bye. Thank
Bye.
you. Thank you. Thank you.
Thank you. Thank you,
Wow.
Catherine.
Good to see
Thank you.
everyone. Miss
Really
you
good.
all. I want to see everyone in person someday.
We have to have a dinner soon.
Someday.